Hi, my name is Gianina Pontrelli, and I'm going to present my poster, which is Glutamatergic Transmission Regulates Locomotory Behavior on a Food Patch in C. elegans. My introduction discusses that glutamate is a neurotransmitter involved in olfaction, which is sense of smell, and decision-making. Disruptions in these pathways can lead to neurological diseases or disorders. Glutamate is a neurotransmitter that is not fully understood, so we just want to learn more about its role as a neurotransmitter. Our specific goals include understanding the normal wild-type behavior in the spontaneous food-leaving assay, identifying the effects of altering glutamate signaling and specific glutamate receptor signals in the spontaneous food-leaving assay and the two non-known dependent food-leaving assay, and finding where in the worm's nervous system the neurons are that are causing the altered behaviors. The first assay is a spontaneous food-leaving assay, where we place the worms on the food patch for 60 minutes and count how many worms leave the food patch. The normal wild-type worm does not leave the food patch. The second assay is the two non-known dependent food-leaving assay, where we place the worms on the food patch and then place a repellent cue right next to the food patch and count how many worms leave the food patch. We then compare the rate at which they leave the food patch to the normal wild-type worms. The normal wild-type worms usually leave the food patch within the first 0 to 15 minutes. For the spontaneous food-leaving assay, we examined the mutant EAT4. EAT4 is a gene that encodes a vesicular glutamate transporter. This helps load glutamate into the vesicles so they can be sent to the synaptic cleft and then be released. Without this gene, the mutant has no glutamatergic transmissioning occurring. So when we looked at it in this assay, we found that it was unable to stay on the food patch and it was all over the plate, which you can see in this graph here. We then did a genomic rescue where we put healthy wild-type EAT4 gene into the EAT4 mutant, and we saw that it behaved like a normal wild-type worm, which tells us that EAT4 is the gene that is involved in this behavior and there are no background mutations. We did a neuron selective rescue where we put healthy EAT4 genes back into the EAT4 mutant, but only in 5 sensory neurons. So there's no glutamatergic transmissioning occurring in the worm except for in the 5 sensory neurons. We found that it behaved like a normal wild-type worm. Therefore, in the future, we can further explore these 5 neurons and 5 exactly which one is causing this behavior, because we know that one of these 5 are causing this behavior. So the EAT4 mutant is unable to smell. That's why it doesn't stay on the food patch. For the two non-known dependent food-leaving assay, we looked at a few mutants. All of these mutants are able to smell. They just decision make differently than the wild-type worm. So the first mutant we looked at was MGL3, which is a metabotropic glutamate receptor. These are also found in humans, and we found that MGL3 is a slow leaver. So we know that this gene is involved in the behavior to leave the food slower. In addition, we looked at GLUR1, GLUR5, which are ionotropic glutamate receptors that are AMPA-like, and we found that this mutant was a slow leaver as well. So we know that these genes are also involved in this behavior. We looked at MGL1, MGL2 double mutant, and we saw no significant difference between the double mutant and the wild-type worm, so we know that these genes are not involved in this behavior. In preliminary data, we looked at AVR14, which is an invertebrate-specific gene, and we found that it is a fast leaver. So in the future, we want to continue to find receptors that are involved in this behavior and find the neurons that are causing this behavior as well. Thank you.